This video will cover the metabolic pathways, which is the sixth part of Unit 1 of Higher Human Biology. This is the first of two videos. This one will cover reactions and the factors that affect them, and also the induced fit theory about enzymes. So the first thing we need to talk about is metabolism. Now, metabolism is often discussed quite conversationally about people having fast or slow metabolism, but as a definition, it is all the chemical reactions within your body, and specifically in your cells. Now, because these are chemical reactions, they occur in the cytoplasm, as with most chemical reactions, and they're also controlled by enzymes. Now, so far, the only term that you've not heard of is metabolism when it comes to National 5. Most of the things that we are going to be covering in this video and in the next refer a lot and will rely a lot on your basic knowledge of National 5 enzymes. So, in metabolism, the pathways can be reversible, which means they can go one way or they can go back the other, or they can be irreversible, which means that they will only follow one path and will not go back the way that they came. Now, there are two types of pathway, which you'll also need to know. Two types of metabolic pathway. One of which is anabolic pathways. Now, in National 5, you will probably have known of this as a synthesis pathway. So a synthesis or a build-up pathway. So because this is a build-up pathway or a synthesis pathway, you would then build up small molecules into large molecules. And because you're building things up, that requires energy. In the diagram above, I've shown a pathway which is reversible. We have a large molecule being changed to small molecules. Or if you look at it the other way, we can have small molecules being built up into large. So this one here is anabolic because we're changing small molecules and we're building up into a large molecule. The other pathway that we have is catabolic. So catabolic pathways are the opposite from anabolic. So therefore, they are the degradation ones we talked about in National 5. So they break down large molecules into much smaller molecules. So in a similar way to respiration, where we break down a larger molecule and we release energy from it, it's the same with catabolic reactions. So we break down large molecules and it breaks them down into smaller molecules. So that then releases energy. So in this diagram above, catabolic would be where you break down large molecules into smaller ones. So this one on the top is your catabolic pathway. So that's your very quick basis of metabolism and metabolic pathways. But now we need to cover what factors affect those pathways and what factors affect the rate of those pathways. So again, like I said before, National 5 knowledge is still important here and we still build on that. So generally, if we think about National 5, we'll know that factors that affect the rate of reaction for enzymes would be temperature and also pH. And with a wee reminder that optimums are involved in that, so enzymes working at their fastest rate, or the conditions that the enzyme are most active at. Alright, the other two main factors we're going to talk about are substrate concentration and end product concentration. So in terms of pushing a chemical reaction or an enzyme controlled reaction in a particular direction, the presence of a substrate would likely push it in one direction, or the presence of an end product would likely push it in another direction, and that means that they would drive the direction call a metabolic pathway. Okay, so it drives the direction of a reaction. So depending on the concentrations of substrate or the concentrations of end product, that would push it in one way or the other. So if we discuss end product concentration first, just because that's the kind of shorter of the two. If you have a high concentration of end product, and what I mean by that is the thing that you're producing at the end of a reaction, okay? So for example, ignoring the anabolic pathway here, if we had a catabolic pathway and it had a large molecule being broken into smaller molecules, the end product would be the smaller molecules, okay, the thing that's produced at the end of it. So when we talk about end product, if they reach a very high critical concentration, then your cells will realise that the molecules they're producing are not being used for anything else, okay, which will be deemed as a kind of waste of resources and a waste of energy or ATP that they'd be using to do that, or even a waste of enzyme use at that point in time. So if end product concentration is high, or it reaches critical concentration, so a certain point, then what will happen is the chemical reaction and the metabolic pathway will have to do something to adjust for that. So what happens is the pathway can either do one of three things. So that means it will either slow down, it might stop, or the reaction might reverse. Okay, so the pathway may reverse. Now the main one we tend to talk about though is not so much about end product, but more about substrate concentration. So I have a diagram under here that I'm going to show you in a wee sec. But generally substrate concentration, okay, substrate, remember from National 5, is what an enzyme works on. So if you have a high concentration of substrate, an enzyme will work fast. Okay, it'll be very active because it has lots of substrate to work on. So if you think back to National 5, if substrate concentration increases, okay, so if you have a high volume or high concentration of substrate, so if it increases, which is what that arrow shows, then the rate of a reaction and the rate of an enzyme controlled reaction will increase. Okay, so the enzyme activity will be higher or greater. Again, back to your National 5 knowledge, the reason why that is because the active sites of an enzyme are coming into contact with the substrate and they're reacting. Okay, so generally that would be the case, substrate concentration increases and then enzyme activity will also increase. But that is only up to a point. Okay, and at that point it levels off. So the activity of an enzyme levels off. I'm going to show you that on this graph here. So your enzyme activity is shown at the side and your substrate concentration is shown along the bottom. So in a graph to show this, what you would tend to see is something like this. Okay, so here in the graph, the substrate concentration is not high enough. Okay, and that's why as you increase it further, then the enzyme becomes more active. So that means that the substrate concentration is limiting, okay, and it is a limiting factor. But the more you give it, the faster that enzyme, or the more active that enzyme becomes. Now at this point it levels off because essentially all the enzyme's active sites are filled. Right, and they are filled with the thing they work on, which is the substrate. So if they're all filled with substrate, that means that they're working at their fastest rate. Okay, and that rate will just stay as it is. Okay, so that's their maximum activity, or their maximum rate. Now you can increase that and bring that level up higher by increasing more active sites. Now you do that then by adding more enzyme. Okay, so you would increase the concentration of enzyme, and that would then add more active sites to it. And then that would allow for that reaction to happen at a faster rate. Okay, so that's a very general introduction to the reactions part. The next part I'm going to do is to cover the induced fit. Okay, so I've got another sheet here. I'm giving a very quick outline of this. Now, when we talk about an induced fit, it's not something that you've heard of from National 5 yet. So it's a higher level point. So it's more complex than the lock-in key that we talked about before, where enzyme action is specific, and that enzyme substrate and the enzyme would fit together. Okay, so to give you a quick overview of what I've got here, this E is representing an enzyme. Okay, and remember the enzymes are made of protein. This area here, an enzyme with a particular shape, is the enzyme's active site. Now, on an enzyme, what you will maybe not have known until higher is that their active site then has molecules on it. Now, if you think about this logically, if enzymes are made of protein, then if you break that down, inside a protein, you have amino acids, and that's what those structures are. So each of these little green things I've shown are amino acids. And those amino acids are found on the surface of that active site. Now the purpose of those is that they have an affinity, right? Which means an attraction for the thing that the enzyme works on, which is your substrate. Okay, so they have an attraction for that substrate and they will pull that substrate in. Okay, so to show you a substrate, remember it would roughly match the shape of the active site. So just for a very quick demonstration, the substrate would look something like that. Okay, and I'll label that substrate. Now that substrate and the amino acids on the active site have an attraction for one another. Okay, so because they're attracted, they'll pull towards one another. And also what these amino acids do, because they have an affinity for it, an attraction for that substrate, it will orientate the substrate correctly. Okay, and what that means is that if the substrate was facing the wrong way, then it would turn it around and pull it in towards the active site so that it's fitting in correctly and in the same or the correct direction. Okay, so it orientates it correctly and draws it into the active site. So the next diagram I have is a very quick outline of that enzyme. And this is where we have the induced fit occurring. Now the induced fit is when an enzyme and the active site of the enzyme changes shape. Okay, so the enzyme's active site changes shape. And it does this to more closely fit the substrate. Now this is very different from what you've known in National 5. Okay, but this is actually a more accurate description of what happens. So if I have a substrate move in to this enzyme, what would happen is the enzyme would then fold or push more closely round about it. Okay, and it would close in on it. Okay, so that enzyme then, the shape would change a bit. If you compare the shape of the enzyme's active site now compared to what it was before, it's different. Okay, not hugely, but it's still different. Right? So that more closely fits to the substrate. Now it does this because it then allows the reaction to happen more quickly. Okay, because it lowers activation energy. Now for the chemist among you, you'll probably know what that means. Okay, for now all you need to know that is it lowers the energy required for that reaction to start. Okay, for that reaction to occur. So it lowers the activation energy, which then will lead to increase in the rate of reaction. Okay, so that means it happens faster. And it does this by stressing the bonds in the substrate. Now, it depends on which type of pathway this is, which depends on whether it would force the substrates together in an anabolic reaction, or whether it would snap those bonds and stress it to break it apart. So in this diagram we have a large substrate molecule, and therefore it's going to be broken down to produce smaller products. So in this example then what would happen is, this substrate, the bonds would become stressed, and that would then break those bonds. It wouldn't force them together because it's not an anabolic reaction. It would break the bonds because it's a catabolic reaction. So then what would happen is, the enzyme would return to normal because it releases its products. Okay, so I'm going to show you the products in a different colour because they are now different molecules. They're different substances entirely. Okay, so these are your products. And they're released from the active site. Now the reason they're released from the active site is because they are not the substrate, and therefore the amino acids do not have an affinity for them. Okay, so we describe that as having a lower affinity. So they have a lower affinity for that enzyme in the active site and the amino acids on it. So they have a lower attraction. Now because they have a lower attraction, then the bonds that would have held them in place break, and then the products are released. So in terms of looking at it overall, you can see that you've got an enzyme, the substrate will bind, and ultimately products are made. Okay, and this example is a catabolic reaction. If it was anabolic, it'd be going in the other direction. So these would then be your substrates. They'd bind together and the bonds would force together through the induced fit. And then you would have this released as a product. Okay, so you could be asked which type of pathway this is depending on which type of question you get. Okay, so this builds on the National 5 knowledge. The next video is then going to talk about inhibition of metabolic pathways.